This video is on factors that impact enzyme activity. We'll start by talking about environmental factors, specifically temperature. When we look at this temperature graph, on the x-axis is temperature increasing, and on the y-axis is enzyme activity. You can see that there's a peak in enzyme activity here, and that is called the optimum temperature for this specific enzyme. And if we kind of drew a dashed line down, I should use a ruler for this, we'd see the optimal temperature for this enzyme's activity. It looks like about 38 degrees Celsius, right? Not every enzyme will have the same optimal temperature, but that's just the optimal temperature of this enzyme. What happens when the temperature is too low, okay? So you can see at 10 degrees, there's really no enzyme activity. And enzyme activity gradually increases as temperature increases until we get to the optimum temperature. We'll label that section of the graph A. So under low temperature, molecular collisions are slower, right? So molecular collisions are slower, and then as temperature increases, reaction rate increases as the enzyme and the substrate collide more frequently until we get to the optimum temperature. Okay. So basically, at low temperatures, molecular collisions are low. But as temperature increases, reaction rate increases, because the rate of the collision between the enzyme and the substrate occurs more frequently. Then we obviously get to our optimum temperature. But then after our optimum temperature, it would make sense that as temperature continued to increase, reaction rate would continue to increase. However, we have to remember that our enzymes are proteins. And as proteins, they're highly affected by temperature, specifically in their tertiary structure. So if we think about this enzyme's shape as the relationship and the bonds between the different amino acids in the structure, as temperature increases, we're putting a lot of kinetic energy into this enzyme, okay, causing these bonds to break, causing the enzyme to lose its form and lose its function, right? So basically, high temperature breaks bonds in the tertiary structure of the enzyme, causing a loss of structure, which causes a loss of function. So we'll label this part of the graph B. That starts to slowly happen and then more rapidly happen until there's no enzyme activity. So B, at high temperatures, break bonds in the tertiary structure of the enzyme. This leads to a loss of structure, which leads to a loss of function. We call this process denaturation. So that's how temperature works. pH has a similar but not the same curve. You can see the temperature, you have a slow increase in reaction rate, an optimum reaction rate at the optimum temperature, and then a fast decline. Whereas pH, it's going to look more like a bell curve, although this graph doesn't look exactly like I'd like it to look, so we'll edit it a little bit. Again, there is an optimum pH at which each enzyme works, and for this specific enzyme, it's 7. That might be amylase. Amylase works at the neutral pH of our mouth. Some enzymes, like pepsin, that work in our stomach, like a slightly acidic pH, actually very acidic pH, like a pH of 3. So their optimum pH graph would peak at 3. Arginase, which works in your small intestines, actually has an optimum pH of more 8, 8.5, so it would peak over here. So these graphs don't always look exactly the same, but it's always a bell curve with pH. So basically, we'll label this. Here, A is acidic, and I'm going to actually draw the rest of this bell curve here. B is basic or alkaline. So basically, sorry, silly joke. If it's too acidic or too basic, right? This also interferes with bonds in the tertiary structure, right? And sometimes in the secondary structure. And once again, this leads to a loss of structure, which leads to a loss of function. And again, we have denaturing occurring, right? Because if it's too acidic, the H plus might interfere, might try to bond with a negatively charged amino acid. If it's too basic, OH minus might try to bond with a positively charged amino acid, and that might affect bonding throughout the structure. Okay. Next, we'll look at concentration. This specific graph is looking at substrate concentration specifically. So the amount of reactant that goes into the enzyme, changing that. If you have 0 substrate, there'll be nothing to go in the enzyme, 0 reaction. If you have 1,000,000 substrate, you might have 1,000,000 reactions. But eventually, there's going to be more substrate than enzymes, so your enzyme will be your limiting factor. So let's look at the substrate concentration graph, right? This really determines the efficiency of the reaction. You can see this part A, right? As substrate concentration increases, rate of reaction increases rapidly. Because all of the enzymes are working. Eventually, though, you reach this like saturation point right here. And that's the point on the graph where you really see this leveling off. Okay? That's because every single enzyme is busy, so even if you increase substrate more, the reaction rate can't increase anymore. So you get this leveling off, this plateau here. Basically, the reaction rate plateaus because all of the enzymes are occupied with substrate. You can't see that. And no increase in reaction will occur unless you add more enzyme. So this is assuming a constant enzyme concentration, and you're just changing the concentration of the substrate. Okay. So some of you all in class did notes on enzyme inhibition, some didn't. So if you did notes on enzyme inhibition, skip this part. However, there is a part on feedback inhibition that you need to fast forward to. But if you didn't do enzyme inhibition yet, here's the notes on enzyme inhibition. Enzyme inhibition is when a molecule prevents the reaction from occurring by changing something in the substrate. So when we look at this, we have kind of separate this into 3 pieces. Okay. In this first example, this is the normal example. Here's your enzyme. Here is your active site. The substrate fits easily into the enzyme, and the reaction proceeds normally. Okay. Sometimes, however, there can be a molecule that has a similar shape, or at least part of the shape is similar to the shape of the substrate, and it competes with the substrate for the active site. So here's the substrate, and here is what's known as a competitive inhibitor. And this competitive inhibitor has a shape that's similar enough to the substrate, at least from one angle, that the competitive inhibitor binds to the active site, right, on the enzyme, and that blocks the substrate from binding. So the substrate cannot bind, okay? And that's going to slow the reaction rate. So in this example, the competitive inhibitor binds to the active site, and this blocks the substrate. Now, most competitive inhibitors don't permanently bind. So this competitive inhibitor can jump out, the substrate can jump in, right? At this point, there's a 50-50 chance of the substrate getting there first or the inhibitor getting there first, right? Let's say, however, I increase it to 2 substrates and 1 inhibitor. Now there's like a 66.6% chance that the substrate will fit in over the inhibitor, so that increases the reaction rate. And then let's say I add 3 substrates. Now there's a 75% chance the substrate will fit in versus the inhibitor. So for competitive inhibition, often this inhibitor can jump in and out, and the substrate can jump in and out as it's broken apart. So they're constantly vying for these active sites. So if you increase the substrate concentration, it does increase the rate of reaction, but not as quickly as in a normal reaction. And we can actually see that over here on the graph. This is, again, our graph of substrate concentration, just like the graph up here of substrate concentration. So this line is showing you as you increase substrate concentration, you increase rate of reaction until it plateaus. That's this curve right here, this normal increase. As you increase substrate concentration, enzyme rate of reaction increases and then plateaus as all the enzymes are full. That's the normal enzyme. In competitive inhibition, right, you start with a slower reaction rate because the inhibitor is blocking the active site of the enzyme. But as you get more substrate, it overwhelms the inhibition, and so reaction rate increases, not as quickly as a normal enzyme. And eventually, if you add enough substrate, if you add 1 billion substrate and 1 inhibitor, the reaction rate is going to look almost exactly like the normal enzyme reaction rate. In our last example, we have something called non-competitive inhibition. In non-competitive inhibition, okay, you still have your substrate that's trying to bind to the active site. Okay, here's the active site right here. But in non-competitive inhibition, okay, you have another site on the enzyme down here. This other site on the enzyme is called the allosteric site. When a non-competitive inhibitor binds to the allosteric site, that pulls on the bonds in this enzyme between the amino acids, changing the shape of the active site. And now the substrate cannot bind. This can also be reversible. So this non-competitive inhibitor can pop out of the allosteric site, and the substrate can bind, and the active site will go back to its normal shape. However, typically this stays for longer, and so it really does slow down the reaction rate. Okay, so let's look at this. Basically, we're going to say the non-competitive inhibitor binds to the allosteric site on the enzyme. And we can see that right here. Okay. That's what happens first. That second changes the shape of the active site. So the substrate can't bind. And again, if you add a lot more substrate, you could outcompete the non-competitive inhibitor, but because they're not both competing for the active site, this really decreases reaction rate a lot more. And you can actually see that on this graph. If this is the normal enzyme, and this is the line as you increase substrate concentration for competitive inhibition, you can see for non-competitive inhibition, the reaction rate is more permanently decreased. Interestingly enough, both examples I shared here happen all the time in the cell to control activity of the cell. And they're not permanent. This competitive inhibitor can pop in and off and on. This non-competitive inhibitor can pop in and out. If you have a competitive or non-competitive inhibitor that permanently binds, that's a poison. Okay, that's like arsenic or heavy metal poisoning or some snake venoms or things like that. Okay, so both of these in the body normally go in and out, but if they don't, then that is considered a poison. The last example I want to talk about is this idea of feedback inhibition. Okay? Feedback inhibition is a way to control the amount of product made in a metabolic pathway. how does it do this? Well, basically, okay, the end product is going to inhibit an early enzyme in the pathway. So let's do this. Let's use as our example here cellular respiration. So our starting substrate here is going to be glucose. Okay. Glucose is going to go into enzyme number 1, and it's going to be broken down into some products. So I'm going to color code all the enzymes the same color. So this is enzyme number 1. Glucose, which is the substrate, is going to fit into enzyme number 1. Okay? And that's going to be broken down into 2 pyruvates. That's not a final product, that's an intermediate in this process. Okay? Here's enzyme number 1 still. Then pyruvate is going to go into enzyme number 2. Here's enzyme number 2. Okay? And it's going to be further broken down in many, many steps. This is too basic. There are thousands of steps, not thousands, hundreds of steps in cellular respiration. Another enzyme will continue to break the sugar down. We'll produce another intermediate. Intermediates are products that aren't final products. They're products of one reaction that act as reactants to the next reaction. So it's going to produce another intermediate product, which is going to fit into this final enzyme. Okay, and this isn't exactly how it works in cellular respiration, but we're just making a point here. Okay, fit into this final enzyme, enzyme 3. Okay, and again, it would be many more steps. And let's say in cellular respiration, well, this is true, one of our final end products is ATP. Again, this is not enough steps, but whatever. So this is the end product. This is the final product. Now, when your body needs to continue to make ATP, then you keep making it. But let's say you've made enough ATP. You want to shut this process down. The most effective way to shut this process down is to shut down enzyme number 1. Because if you shut down enzyme number 3, you're still doing the steps for enzyme that the reaction enzyme 1 is catalyzing and the reaction enzyme 2 is catalyzing, right? That's wasting a lot of energy. But if you go all the way back and shut down this first enzyme, this whole process is shut down. So let's say you've made enough ATP. Well, that ATP can go back up here and act, the bound end product, the ATP, can act as a non-competitive inhibitor to shut down this process. Okay? So again, that's called feedback inhibition. What's happening there is the final product inhibits an early enzyme in the process. Right? This is also an example, and then because if you shut this down, then you won't make more ATP. Now, when you run out of ATP, this ATP can come off this enzyme. This enzyme can be reactivated, and the process can start again. So it can go back and forth and back and forth and back and forth. So I would be remiss in not telling you, this is a great example of negative feedback. Negative feedback is when basically the opposite result. Whatever you end with, the result is opposite. So let me give you an example. So for example, if you increase ATP production, then you inhibit the process to stop ATP production. So you start with too much ATP and your end result is stopping ATP production. Or you don't have enough ATP, you activate the process to start ATP production. Both of these are examples of negative feedback, right? If you have, like if this was on the graph and this was like ATP and this was like time, right? And then, okay, you're producing more ATP and then you have too much, so you shut it down. And then you don't have enough ATP, so you turn the process on. And then you have too much ATP, so you shut the process off. This is negative feedback. Both of these things are negative feedback because the results and then what you end up doing are opposite processes. Okay? And this really helps to maintain a very important thing, which is this helps maintain homeostasis. Because the stimulus and response are opposite. Let me zoom in a little bit. And this happens all throughout our body, but this is just a good first example of negative feedback. I hope you enjoyed my video on enzyme.